Thank you, Bill. And it's been a pleasure with Bill and one of the Bryan Green Woods in this case to help organize this. I think it's been a fantastic day. And as we come to this final session, I think one of the things that we really wanted to do was kind of look to the future. I think enormous progress has been made using the tools we have now. But I think for many of us anyway, myself included, it is this discovery of new things about the disease, new approaches, new ways of thinking, new strategies that I think are going to get us over the finish line. And so I am delighted that we have such a good group of speakers for this next session. The first is the other Bryan Greenhouse from the University of California at San Francisco, who's going to talk about tracking malaria from a drop of blood. Bryan.
Thanks, Diane, and thanks for the organizers for inviting me to such a fun conference. So while they're getting this up, we're going to do a little audience participation. Who likes data? Put your hands up if you like data. Good, this is not a trick question. Correct answer. All right, who likes passing up incredible opportunities and wasting a lot of money? Put your hands up. Okay, great. You guys are the right audience for this talk. All right, thank you. So I'm going to talk about tracking malaria from a drop of blood, and that title will become a little bit less cryptic in a minute. This is my motivation slide. I don't think I have to spend too much time on this after all the discussions that we've been having. But basically, elimination is going to be really expensive. Okay, and doing it in a half-hearted way is not going to be enough, as illustrated on the right there. A really nice paper by Justin Cohen et al., where they've showed in a number of different settings when you relax things or stop things in terms of interventions too soon, malaria comes back and it can be devastating, right? So we can't do everything everywhere, but we need to do enough in enough places. And that's really the balance that we have to strike. So in order to do this right, we need to be smart about which interventions we use and where. And to do that, we need data, right? And the better our data, the better we can make rational decisions and get this balance right. All right, so this is how we track malaria now. Basically, I'm going to distill this down into three main ways. So entomologic surveillance, very important. Probably not something that we're going to do at scale to measure transmission per se, and certainly not in very low transmission areas where we're thinking about elimination. Right, so I'm not going to talk about entomologic surveillance for the rest of this talk. So that leaves us with two things. Active surveillance, okay, where we go out and we spend lots of money to go and test people in communities or in schools or using snowball sampling or some way we're just going out and testing people, right? And they're going to either be positive for malaria parasites as indicated by having a red drop of blood, or they're going to be negative by whatever diagnostic test we're using as indicated by a hollow drop of blood, right? Or we do this by passive surveillance, where people come to us seeking care, right? And there's many different flavors of both of these things. So at the end of the day, this is essentially what we get, right? Or if we're talking about a place that's close to elimination, it's probably a little bit more like this. We get counts of positives and negatives. So if we did a survey, we just take the total number of people we assayed, and we look at how many are positives, and we get parasite prevalence or parasite rate, right? If we're doing this in the setting of passive case detection at a health facility, we also get a prevalence. We call it test positivity rate or something like that, but it's essentially the same thing. It's the percentage of people that are positive. If we're feeling really ambitious and we feel comfortable making a lot of assumptions, we can try to estimate incidence of malaria and possibly other things, all right? But that's basically where we're at. So these measurements are very useful. They're the basis for nearly all of the data that we have right now on malaria transmission, but there's a lot more hidden in each one of these drops of blood if we know where to look. So all of those negative drops are from people that are not infected now, presumably, but many of them have been infected in the recent past, right? And so their blood, if we look closely enough, contains a log of these previous infections, sort of like a visitor's book at a hotel, which allows us to see into the past and get a lot more information. The ones that are positive contain an additional treat, okay? Not only a record of parasites that have visited that person, but the parasites themselves, each of which carries essentially a passport that's stamped with all the places that it's been over the years. Right? And of course, if I haven't seen any fluorescent microscopy results that look exactly like this, so we need to do something where we're translating antibodies into these visitors logs, right, and DNA from the parasites into these passports that are stamped. And I just wanted to point out that remember, we're already spending all this money and effort getting these drops of blood. So, and it's really trivial to save a couple extra drops on a piece of filter paper or even just used RDTs, which is another source of parasite DNA material. So in order for these techniques to be useful, we don't need to get this perfect. We just have to do better or add value to what we're already doing. All right, so let's start with making these visitors logs. So here's a parasite survey. Here, instead of a drop of blood being read, there's one individual who's infected in this particular survey. If we look back over time, which we can do with antibodies, we can see that maybe some of these individuals are not infected now, but they've been infected in the past. And just to echo something that Rick said, this is in the session for new tools, but antibodies are actually a really old tool. They've been used since at least the 1960s, looking at immunofluorescent assays. Now we have better tools. We can use purified recombinant proteins and ELISAs or other techniques to measure antibodies. But still, most of the antibody work that has been done to look at exposure have been looking at responses to a few highly immunogenic antigens, which essentially means, hey, somebody's visited you at some point in the past. You don't necessarily know when or for how long, and those are potentially important pieces of information. So we can't necessarily distinguish recent from distant exposure with a lot of those antibodies. However, they're still very useful. We can make a distinction because, for instance, we know a 3-year-old can't possibly have been infected 5 years ago, right? So if we do an age stratified sample, we basically, whatever way we're getting blood from people, we make sure that we have different age groups included. And we can look at the parasite antibodies that are present in these individuals by age, and we can look at how fast people acquire these antibodies and estimate transmission at the community level. So this is really kind of version 1.0 of this visitors log. It's not really at an individual basis, but it's looking at the community level. So this works really well if transmission's been stable. And I have on the left side there an example from 3 sites in Uganda from our first isomer study, where we had 3 sites where we did really detailed entomological surveillance and cohort studies. And those differences in transmission are really reflected very nicely by the rate at which these people develop antibodies to a very long-lived antigen. So the green curve is showing Nagangara, which Toon has already talked about a little bit. EIR there used to be 300. And this was taken in the setting of very high transmission. Chihihi, where it was about 30, and Welakuba, where it was about 3. And you can see that those curves plateau at different heights, and they kind of arc in a different way. So we can calculate something called the seroconversion rate, which reflects this transmission intensity. I'm just showing you an example here from 3 sites in Uganda, but how does this work more broadly? So here's an example showing data from areas where we have parasite prevalence, entomological inoculation rate, and antibodies, which I'll show you in a second. And I'll just point out that with looking at the parasite prevalence, you do see a relationship, right? This is, there's a relationship here. But if you were to try to estimate, say, the EIR from the parasite rate, you can see that there's a factor of about 100 here over which EIR varies for basically the same parasite rate. And that's true all the way up and down the curve. So we can get some rough idea of transmission intensity, but it's not necessarily as precise as we'd like. From those same exact samples, measuring measuring antibodies from dried blood spots from those same individuals, I'm now showing points of gray, looking at the seroconversion rate calculated for each of those communities using a single antibody. And you can see it's a much tighter relationship, and if you just look at the R-squared, it's about double. So we can already, just by using kind of a single antibody that's long-lived, from the same survey that we spent maybe millions of dollars to conduct, get a lot more information on transmission. And just to say that even though it's a bit of a blunt instrument for looking at changes in transmission over time, we can extract this information to some degree in areas where those changes in transmission have been very dramatic. So here's a recent study that's been published by Anders Bjorkman et al. And they showed basically by looking at how this curve shifts, sorry, wrong button. Right there, you can see that basically over the last 10 to 15 years, transmission has been at a very different level than before that. All right. So we can do better than this, especially in this setting where we're trying to actively drive transmission down, right? I said this particular tool is probably very useful and most useful in settings where transmission has been relatively stable. How do we do this? Well, The nice thing about malaria, and also one of the things that makes it very difficult, is that it's a very large parasite in terms of its genome. So it's got about 5,000 different proteins, and we're only looking at antibody responses to 1 or 2. But presumably, these different proteins have differing degrees of immunogenicity. And if we can combine data from antibodies with different kinetics, we can try to reconstruct some of this information. So here I'm just showing a cartoon on the left where you, sorry, it's your right, where you see a few different people that have been infected at different rates over the past. Looking at one antibody, they basically all look the same. But if we were just to add a second theoretical antibody here with a much faster kinetics, you can see that they would potentially look different. We might be able to distinguish the person on the top who's had some historical exposure but nothing recent with the person on the bottom who's had some degree of recent exposure as well. So how do we go about building these? We're calling them cameras, and I'll show you why in just a second, which is really version 2.0 of this visitor's log. We're calling them cameras because it's a very obvious extrapolation from combining antibodies to measure exposure recency assays. And the schematic is a bit busy, but I think I'll just walk you through it very quickly, how we can go about doing this. So essentially, we start with really good cohorts. These are expensive studies, probably designed for other reasons, where we can go back in time and see when people have been exposed. We then measure a whole bunch of antibodies for candidate selection, and we can do that either using a bead-based array or protein microarray, or now we're actually developing some phage array approaches to look at hundreds of thousands of different targets at the same time. You put them all into a bucket, you have your data, you're on exposure, you have your data on the antibodies, you use some sort of statistical methods to select the ones that are the most informative, you do some additional validation, and then ideally you design some very targeted assay that's maybe looking at a handful of different antibody responses that gives you very precise information about the past. So how do we know this will work? Well, we've done it. We've done it in Uganda. We published a few years ago that we can take 6 antibodies from at one cross-sectional time point and do at least as good a job as a full year of incidence data in a cohort. So I'm showing, again, data on the left based on 6 antibodies and on the right based on parasite prevalence for the exact same populations. And you get a much better dynamic range and greater precision by using 6 antibodies than by just looking at one point in time if someone's parasite positive. All right, we've also defined use cases for elimination. I'm not going to go into these in detail, but just know that a recent paper came out from as a consensus document. And we have defined these, especially for the elimination context, but I will point out that even though this kind of ends at low on the left here, stratification of risk and measuring changes due to interventions really extends all the way to the very highest transmission. And I'll just end this section by saying that we're working on, in a very collaborative way, developing cameras that are much more generalizable than the ones we originally developed in Uganda, which I really view just as a pilot study. So we're looking at a wide range of cohorts indicated on that map by different colored circles with many collaborators, all of whom have very high quality cohorts where we can look back in time at exposure. We're doing some de novo high throughput antibody screens, looking not at just a total IgG, but different subclasses in IgM, which may be very informative as well. And then the eventual goal is to really have an assay or set of assays that you can very robustly apply across a wide variety of ages and transmission settings. All right, so that's it for section 1. Now let's talk about the second set of tools, turning DNA into passports. And to motivate this, I'm not going to go into too much of the biology, but just to say that Plasmodium falciparum is an extremely diverse parasite on the genetic level, and that diversity is maintained by sexual recombination in the mosquito primarily. So this is really at a time scale that's very relevant for transmission because it only happens once with every transmission event. I'll also point out that especially in sub-Saharan Africa, where I do the most of my work, infection with multiple strains is common, and not only common, it's really the rule rather than an exception. That makes certain aspects more challenging, but it also is an opportunity because those combinations of infecting parasites are often unique. And if we start thinking about this as a combination of parasites that are infecting someone as opposed to trying to think about it at an individual parasite level, we can do a much better job about reconstructing transmission. So on the right side there, I've got a schematic, very simplified schematic of what a transmission network might look like. Each of these, it's important to know that each of these circles is not a parasite, but a person who may be infected with multiple different parasites. The dashed circles indicate the fact that we know we're going to be missing chains in this infection. We're not going to get everybody. And ideally, you would do this in such a way that you're not just using the genetic data, but you're incorporating all the other things that you have at your fingertips. So temporal data, spatial data, including GPS coordinates if you have that information. All right, and this is just to indicate that it's never going to look quite like that stylized diagram on the right, but a little bit more like what we have on the left here. It's hard. So we're not going to do it perfectly, but remember the goal is not to do it perfectly, but to add value to what we've already done. All right, so I'm just going to motivate this with a couple of quick examples, and I'm not going to go into too much detail, but hopefully leave time for questions if people have specific questions about the methods or more data. So here's one example from the Kingdom of Eswatini, formerly known as Swaziland, and they've almost eliminated malaria, right? So I've shown on the left, bottom left here, you can see the cases per year reported. These are confirmed cases reported by the National Malaria Control Program. You see there was a major drop, but then, as Rick mentioned, this is true in many other areas, kind of stuttered around near this baseline for many years, right? So some important questions are, how many of these cases are acquired locally versus imported from somewhere else? Ideally, we'd like to know where the imported cases are coming from, so we could potentially direct some cross-border coordination of interventions. We'd like to know of the local transmission that we do observe, when and where does it occur, and how sustained is it? And related to that, what are the reservoirs and drivers of local transmission? Right? So fortunately, Eswatini has an extremely robust surveillance system. Right? They've collected these data over 10 years. They actually have GPS coordinates of most of the people that have cases. They ask them extensive travel histories. But as Amy pointed out earlier, that's not necessarily enough data. So in red here, I'm just showing the number of cases that are local as defined by travel history. All right, although these may be local or they may be introduced, which is the first generation of a locally transmitted case. Amy already showed this slide, but I just want to quickly point out one additional point. So the reason that travel history is inadequate is not just because people may not tell you the truth, or even if they tell you the truth, you may not be able to determine where they're coming from. It's also because even if you had perfect characterization of every single case, as whether that was acquired locally or not, there's a whole problem of missing infections, right? And there's been some attempts made to estimate transmission intensity based on the proportion of cases that are local versus imported based on travel history. But as indicated by these two cartoon diagrams on the left and the right, they both have the same number of local cases, and they both have the same number of imported cases. But the situation on the left is very different. You've got very limited local transmission. You've got mostly dead-end chains. And on the right, even with the same case counts of both local and imported perfectly characterized, you've got a very different situation where you've got sustained local transmission. Okay, so this is kind of be my methods slide. It's very rudimentary. We go out, we get dried blood spots. In Eswatini, we have the advantage of both doing this through reactive case detection and passive case detection. So we have some asymptomatics as well as symptomatic cases. We do some sort of multi-allelic, multi-locus genotyping. We've historically done this with microsatellites. Now we're doing this with a panel of microhaplotypes that we're deep sequencing. We then identify the alleles that are present. And in the situation, even here in Eswatini, we usually have multiple alleles at each locus. We use some statistical methods, and we combine that with epi, and we try to reconstruct as well as possible the transmission. Okay, so here's a little bit of data. So local transmission is really, really rare in Eswatini. So this is good news. And we know this with a pretty high degree of confidence because we genotyped about 1,000 infections over 3 years, which is about 2/3 of all the cases reported in the country. But I will point out that although most of these chains are really short, and in fact, it's hard to even call them a chain, they're just isolated nodes, it's very heterogeneous. You do have some cases of sustained transmission, and so we can really look when and where those are occurring. I'll also point out, I know this has been discussed quite a bit today, that at least in this particular instance, we do see that asymptomatics contribute to local transmission as much or more as the symptomatic cases. And again, this may have in part to do with the rapidity with which transmission comes down, right? And that's not, Kim mentioned that for communities that are sort of isolated, but in this case, it may have to do with not just the rapidity with which transmission has been decreased locally, but how much of a migrant population you have there coming in and what the immunity level is in those people. So we have a lot of migrant workers coming from Mozambique. They may be very immune, and it's likely a lot of those people that are the asymptomatics contributing to transmission. So in these kind of border areas or areas of high mobility, it's hard to characterize necessarily the local population, both of people and of parasites, because there's so much intermixing. So it does confuse things quite a bit. Another really important thing is that there's almost no transmission from one season to the next. So there doesn't seem to be some smoking gun, hidden reservoir in Eswatini that's really seeding transmission. It's all seemingly due to importation. Some words disappeared from the slide. But most local cases are actually probably introduced cases in Eswatini. So again, an introduced case is very important to distinguish from a local case, because local implies at least 2 generations of transmission. And at least in the older version of the guidelines, there was a pretty important distinction for elimination. Maybe Bryan can comment later on what the newer guidelines are going to look like. But it's important to know that of these 50% of the cases in Eswatini that were local, almost all of them seem to have been kind of a dead end, probably a one-off from an imported case that we couldn't find because they were probably asymptomatic, and we would have never captured them through standard epidemiologic surveillance. The other thing that's really, again, a positive thing for the country is that there was a substantial number of cases that had unknown travel history, and this is because their strategy is not to ask travel history at the health facility, but to go out later, and so they miss a lot of those people. And almost all of those genetically look much more like the imported or introduced cases than the local cases. So that's another piece of good news that you wouldn't know just by looking at travel history. And I'll also point out that these passports are very useful domestically as well as internationally. So we can not only look at local and imported, but again, of the locally transmitted cases, where and when are they occurring? They're mostly occurring over a few kilometers, as you'd expect, but there is a long tail. All right, just a couple more slides. So at larger scales, we can also evaluate connectivity between populations of parasites transmitted, not just individuals. And Amy Wazowski already showed a number of slides on this, but just to show one more, this is from the same study where we collected used RDTs and dried blood spots in the border areas of Namibia. And we found much more detailed and extensive evidence of cross-border importation, or as Kim suggested, maybe importation is the wrong word, but kind of a cross or on the border focus that really spans the border, than the sparse travel history data, right? And this is because even though we use the same population of people, in fact, we had more people for travel history than we did for genetic data because we didn't genotype every one of them. But each parasite carries with it not just where that person came from, but where the transmission cycle before and the cycle before that came from. So you can pull that information a little further back into the past than just the individual that you're asking. The parasite tells a story that goes back a little further. All right, so take-home messages. Surveillance is critical. We all know that. It's very expensive. And I'm going to make the argument that for a marginal increase in cost, in the right settings, we can get much more accurate and detailed data on transmission if we use antibodies and parasite genomics in a rational way. There are many areas that are active for development. Amy touched on this a bit before. We're constantly improving laboratory methods, the antigenic targets for antibodies and the genomic targets for looking at the genetics. I think probably the biggest area of need and also a very exciting area of development right now are the analytics to not just look at kind of relative flows, but ideally calibrated epidemiologically relevant metrics. So for instance, what is the force of infection? What is the basic reproductive number? What are the migration rates of infected people from area X to Y? And I think once we get that right, we're going to need to really think about how we best integrate this with other surveillance data modeling and ultimately decision making. And just to point out that at least this is a very biased opinion, obviously, but I feel like molecular surveillance is a very exciting field for research and also implementation science with highly relevant results. And with that, I'll just thank many people that were involved, including my volunteer illustrators. And I'll take some questions. Thank you very much.
Bryan, for a very exciting talk. We have time for a couple of questions. Yes, please identify yourself.
Oh, hi, I'm Christine Hopp. I'm coming from the NIH. I was wondering, have you tried, I don't know if it's possible, but can you combine the passport information with the guest book information and from that learn about longevity of B-cell responses? Because obviously that's a big question. How long do malaria-specific B-cells actually stick around?
Yeah, that's a great question. So those questions I feel like are a bit easier to identify in settings where you've got more precise information about an individual's history. You have to tease apart a lot less. I think probably once we have kind of the camera is worked out a bit more and we can say something a bit more precisely, I think they'll be very useful tools for studying immunologic responses because we'll have a set of information about exposure that's hopefully in some ways orthogonal to whatever we're looking at in terms of immune responses that are different. But I think using cohorts or other detailed studies in areas where we know transmission has changed dramatically in one way or the other might be in some ways a more solid way because we're making less assumptions about that. I don't know if I answered your question, but we can talk more later if not.
Hi, Bryan. Thank you. That was great. Really appreciate it. So I always ask this question of Chris Drakely, so now I can ask you too. So in terms of the serology, it's really exciting stuff, and it seems like it's come quite a ways. I'm wondering if you think now that the serological tools that folks are using, your group and other groups, is there enough standardization between groups and how these are done and measured and so forth that we can begin to understand and compare across some of these sites?
Yeah, Chris might be more optimistic. I would say the answer is currently no, but hopefully in the next few years, we're really working towards some limited set of targets that are very informative. We've even got a prototype like RDT type test looking at antibody responses. I think it's based on a single antibody. I don't think a single antibody is going to cut it, but probably something like 5 or 6. So the technology will have to be slightly different, but we're not talking about hundreds. We're looking at thousands now just to look at candidates. But I think probably in the next, say, 2 to 3 years, we'll hopefully have enough data that we can really figure out that trade-off between complexity of the assay and the information that we want. And then obviously there'll have to be some tech development on a deployable assay. Yeah, ask me in a year or two.
Hi, thank you for your talk. I'm Mariko from Columbia University. I might have missed this in your presentation, but I was wondering how did you determine which genes you were looking at for your microsatellite haplotyping?
That's a great question. So we just did it because it was easy. We used 10 microsatellites that have been around forever that Tim Anderson used back in the day, and then we developed assays that were kind of flanking them. But we're really moving away. So we have a multiplex PCR that works really well for very low parasite densities. We've moved away from it just because it's so difficult to implement in a standardized way across labs. And we're moving more and it's so much easier and cheaper to do sequencing if you do it in kind of a rational and targeted way. So we're working towards deep sequencing of microhaplotypes that have many different alleles. So you get the multi-allelic benefit of really seeing what's in a multiclonal infection that you would with a microsatellite, but it's just much easier.
Right, and maybe a follow-up on that. Do you think standardization is more possible with the nucleic acid assays?
Yeah, this is a question that always comes up, and I think to some degree, yes. But another, I just had this discussion with Amy Taylor last week. The degree to which or the place at which that standardization needs to occur between what you're measuring and your epidemiologic outputs could happen at multiple different places, right? So it could be that you have some analytical approach for looking at genetic relatedness, or even what the next level is in terms of kind of transmission intensity. And if you can standardize your methods at that stage, then it's almost irrelevant what targets you're using because they might be different in different settings. In Southeast Asia, you might need to look at a completely different set of targets to get the same exact information that you would in Uganda.
Right.
So I think there's probably a role for some standardized assays, at least to make things easier in the community, but I think that's not necessarily an essential point to still get very standardized data out of the assays.
Right, and it seems to me actually it's the methodological standardization
Yes.
versus the...
Yeah, the analytical methodology. Yeah,
right.
yeah.
Okay.
I agree.
Great. Well, thank you, Bryan.
Great, thank you.